In the previous video, we have learned about the plasma membrane of the bacteria, and the another one which is most important in the cell envelope, that is known as cell wall. Apart from this, the plasma membrane, cell wall is another major constituent of the cell envelope, which have several functions, like it helps to determine the cell structure, protect the cell from osmotic pressure, then also protect them from various toxic elements, etc. So these are the basic functions of the cell wall. Now, this cell wall is basically made up of sugar moieties and also the amino acid moieties. There are several identical subunits which are present in the cell wall, and each of the subunit is made up of two sugar moieties or sugar derivatives. One is N-acetyl muramic acid, and the another one is N-acetyl glucosamine. Now the short form of this, our N-acetyl glucosamine, is known as NAM, and N-acetyl glucosamine is known as NAG, and N-acetyl muramic acid is known as NAM. So NAM, NAG. So these two sugar moieties are alternatively present in the cell wall like this. So this is the basic sugar moieties which are present. Now, from the NAM, the carboxyl residue which is present in this sugar is linked with L-alanine amino acid. So there are total 4 amino acids which will make a short peptide, and the first one is L-alanine, which will cross-linked with this carboxyl moiety of NAM, and then the second one is, this one is the first, second one is D-glutamic acid, number 3 is L-lysine in case of gram-positive bacteria, and the fourth one is D-alanine. Okay, so this is a sugar moiety, and this is the peptide which is made up of 4 amino acids. So these are generally present in a single subunit. Now from the other subunit, that also contain the similar structure. Like this, I should erase it and rewrite this. NAM, NAG, NAM, L-alanine, D-glutamic or glutamic acid, then L-lysine, D-alanine. Now, on the other hand, the same is present in the other residue like NAM, NAG, and NAM, like this. So from this NAM, there is also L-alanine, D-glutamic acid, L-lysine, and D-alanine. So there is a connection or a cross-linking between this third L-lysine with the fourth D-alanine with the help of 5 glycine residues, which are known as actually pentaglycine bridge. So with the help of this 5 glycine residues, the L-lysine at the third position from one subunit will cross-link with the D-alanine at the fourth position of another NAM-NAG residue of the another subunit. So this is the basic of the cross-linking, initial cross-linking between the subunits, and each subunit like this will make a peptidoglycan strand, and each peptidoglycan strand then cross-linked with each other to form a thick mesh-like peptidoglycan polymer, which is the major constituent of the cell wall. So PDG is the major constituent of cell wall. It's basically a mesh-like polymer present in this cell wall. Now in case of gram-positive bacteria, this PDG layer is about 20 to 80 nanometer thick. So there is, in case of gram-positive bacteria, there is a PDG layer, like this, and a plasma membrane. Like this. And in between the PDG layer and the plasma membrane, this space is known as periplasmic space. So this is the whole cell wall and plasma membrane structure of gram-positive bacteria, in which there is no other layer present, and this is the complete structure of the cell envelope which is present in case of the gram-positive bacteria. Now, apart from this, in case of gram-negative bacteria, there is also a peptidoglycan layer, but that is very thin, which is about 2 to 7 nanometer thick, and outside of this PDG layer there is an outer membrane portion which is about 7 to 8 nanometer thick. So in case of gram-negative bacteria, there is an outer membrane, there is a PDG, and then finally a cell membrane. And in between these places, these are known as the periplasmic space, which is much more similar as the gram-positive bacteria, though the structural variations can be seen between the both gram-positive and gram-negative bacteria. So this is the basic structural analysis of the cell wall in case of both the gram-positive and negative bacteria. The most important thing is, in case of gram-negative bacteria, the PDG layer that contains, again, the NAG residues, like the gram-positive one. But here, from the NAM, the first one is similar like those the gram-positive bacteria, that is L-alanine. Then second one is D-glutamic acid. Third one is there is a diamino acid always present in case of the gram-negative bacteria. In case of gram-negative bacteria, the third position is substituted with diamino acid. It is not similar with the gram-positive one. That is known as mesodiamino pimelic acid, or in short it is known as DAP in the third position. The third position, that is mesodiamino pimelic acid. The fourth one is again D-alanine, and the same it is also here, that is L-ala, D-glutamic acid, mesodap and D-alanine. So basically this D-alanine is cross-linked with the mesodap. The amino acid present in this peptide at the fourth position, that is D-alanine, it is always cross-linked with the mesodiamino-pimelic acid, which is present at the third position in the another NAMNAG residue moiety, and in this way they will cross-link with each other and make a PDG strand, and each of the PDG strand will again cross-linked with each other to make a complete peptidoglycan layer. But yes, in this case this layer is much more thin than as compared to the gram-positive one. So these are the basic differences between the cell wall structure between the gram-positive and negative bacteria, because both of the bacteria have the PDG, and most of the bacteria are divided into the two major groups according to their response to the gram staining analysis. So this is it. Thank you for watching this video.